Monday tissues. If there's some extra time on Monday, we might do a little review. We might start talking about anatomical terminology. But if we do anatomical terms, it will not be on the first exam. It will be on the second exam. But we might start that. It depends on how far we get today and how fast the pacing is on Monday. We're a little bit behind in the course. Not too bad, though. You will have a quiz on Monday. It will cover tissues. We just started tissues a little bit on Wednesday, and then we will continue them today as well. So it'll cover up through whatever we get through the day on the quiz on Monday. 10 points beginning of the time period, same format as before. Your first exam is Wednesday of next week. It will consist of 50 multiple choice questions. We'll be using Scantron forms. You'll need to bring a pencil. So if you don't normally bring a pencil, bring a pencil on Wednesday. And then 1 to 2 pages of short answer. By short answer, I mean the kinds of questions that you've done so far. It might be something like this right here, or maybe diagrams where you have to draw an arrow through or add something a little bit, or you can answer in a couple of words, but not long essay, not something where you have to write 5 or 6 sentences and explain. Maybe 1 or 2 sentences to maybe explain something, but not long essay anyway. Short answer, fill in the blank, labeling diagrams, answering something you see about a diagram, and so on. I'm going to have a review session on Monday. I just don't have any time on Tuesday to do it, otherwise I would do it on Tuesday. Tuesday's my busy day. A review session on Monday afternoon in this room from 4 to 5 o'clock. I'm not going to have anything formal planned. Well, probably not. I might have a couple things. But you guys are going to bring in whoever wants to come, and this is optional, this is voluntary. Come in and bring questions for me to answer. If you bring in a written question, I'll give you 1 bonus point on your exam. So bring in a written question, just a piece of paper, 3x5 card, whatever. Somewhat relevant, at least to the topic being discussed. It could be something directly relevant, something that's likely or could appear on an exam. It could be something that you just have an interest to. I don't know, you've got a disease disorder that's related to something that we've talked about. I'll try to answer it. I might not be able to get all of those. We'll see. But again, bring a written question in on the review session, 4 to 5 o'clock, and you get a bonus point. If you've got a class that runs up to 4:10 and can't get here a few minutes late, that's perfectly fine. If you've got a class that starts at 4:30 and you have to leave a little bit early, that's fine as well. Again, we'd like you to be here as much as possible because the benefit from being here at the review session is it's not the idea of submitting the question. The benefit is you hear all the different questions, answers from a lot of different people. Any questions about any of the announcements, previous material, the review session of the exam? Oh, I must have went through those fairly good then, so there were no questions on that. And that means we are ready to continue tissues. I believe this is where we left off last time. We had, actually I'll go back 1 slide. We had already mentioned that there were 4 general categories of tissues. We're going to get through epithelial today and hopefully start a little bit of connective. And then we'll finish the rest of them on Monday. Epithelial will take a little while to get through. We left off with this with some general characteristics about epithelial tissues. Let's see if you remember something that we talked about last time. What is matrix? This is just a reminder from what we talked about last time. What was the word matrix? What did that mean? So, matrix referring to blood supply. This says epithelial tissues have little matrix. They have little blood supply, little cytoskeleton, little cytosol, a little extracellular material. What is that? Alrighty, let's see what you guys remember from last time. And pretty good. If you chose letter D, you are correct on this. Remember, matrix is extracellular material. And if we go back and look at these cells, notice they're all tightly packed together. That has some important consequences when delivering drugs. We'll talk about the pharmaceutical importances of connective versus epithelial tissues maybe a little bit later. But that has some important consequences with that. Okay, get out your drawing hands and drawing fingers. We're going to make some drawings. about how we classify epithelial tissues. We're getting ready to look at different kinds of epithelial tissues. Epithelial tissues are categorized based on two things. Number one, shape of cells. Number two, layering. What do we mean by that? First of all, shape of cells. Sometimes cells may look a little bit like this. Okay, how do I want to draw this? That's supposed to be a flat cell, and epithelia that have flat cells are called squamous epithelia, or the cells are called squamous cells. Squama means scale-like. You might think of a fish's scale as being kind of a flattened structure. So I think that's the idea of squamous. So an epithelium composed of flat cells is called a squamous epithelium. Sometimes the cells look like this. I know I'm not much of an artist, so that makes it easy on you guys. If I can do it, then you guys can draw as well. That's supposed to be a cube. It looks like I got it a little bit off. Okay, there's a better cube shape. Sometimes the cells are rather cube-shaped, more or less, with equal sides, and we'll call those cells cuboidal, and epithelia that have those, cuboidal epithelia. Third and last kind of shape that we recognize are very tall column-shaped cells. We'll call these cells, these tall ones, look like building skyscrapers. We'll call them columnar cells. And sometimes they can be very skinny and tall. They can be really skinny, very elongated cells. We'll call those columnar cells, or tissues bearing those kind of cells, columnar epithelia. So now we know one of the two categories in the way we categorize epithelium. One is by cell shape. The second is by cell layering. And the way we want to do this is I'm going to show you a couple of pictures in the same orientation that you're looking at right here. So we're looking at a sectional view. So imagine an epithelium as a sheet. So imagine this epithelium as a sheet. You can look at the surface of it, you can look at the edge of it. We're going to look at the edge of it. That's what this is showing. Imagine this as you're looking at the edge of a big sheet of cells. That's the orientation that we're going to be looking at. Okay, so first one we're going to be drawing is going to take me a few minutes to do. So I'm drawing our epithelium. That's the cube-shaped cells here. That's the actual epithelium. Again, it connects to some sort of an underlying tissue beneath it. There is a space on the other side. That may be an air-filled space, liquid-filled space. Could be facing to the outside of the body. Could be facing the inside of a bronchial tube, inside the gut, surface of an organ on the gut, facing inside a blood vessel. Lots of different spaces. Underneath it, there is something called a basement membrane. Basement. Which helps to anchor it to the underlying tissue. We can see that over here by these dark blue bands. We can identify a surface that faces the space called an apical surface. In contrast, the surface that touches the basement membrane or that connects to the underlying tissue, we'll call that the basal surface. So just some terms to get oriented. So what we've drawn is the edge of a transection or transverse view of an epithelium. The apical surface is now facing up toward the space. Basal surface is down toward the connecting tissue. It's connected to it by a basement membrane. And what we see is a single layer of cells. Now we get into our category by layering. This is a single layer of cells. It's a different color. We're kind of running out of room here. We'll call this a simple epithelium. And because we have cube-shaped cells, we can now put both of these together, and we can call it a simple cuboidal epithelium. So our top row is how we categorize epithelia by shape of the cells. The second row across the bottom of this is how we categorize epithelia by the layering. And we see it's a single layer of cells, and the cells are cube-shaped. So we're going to call it a simple cuboidal epithelium. Questions so far? Yes?
What's the difference between the basal surface and the basement membrane?
The basement membrane is the actual fibrous material right here that actually anchors, I guess, that anchors it to the underlying connective tissue. The basal surface would be just the bottom of the epithelial cells. So if you imagine this edge up here would be the apical surface, this edge right down here would be the basal surface. I'm going to have to erase one word just to give myself a little bit more working room, and we will draw the next kind of layering in our space right here. And we're going to show the same kind of orientation. So I'm going to put in a basement membrane. We'll abbreviate that, basement membrane. We're going to have our apical surface with the space facing up, same as before. Our other tissue, what it connects to, is under here, same orientation before. And now we're going to do something a little different with the layering. So I'm not going to bother to put in nuclei with these. But each block is supposed to represent a cell. I mean, I could put nuclei in these if we wanted to, but we don't have to. What we see now is 3 rows of cells, 3 layers of cells. 1, 2, 3. Anytime that we have more than one layer of cells, whether it's 2, 3, 20, 200 layers, and some of them might approach 200, some epithelia, as long as it has more than one layer of cells, we're going to call it a stratified epithelium. So this one is a stratified epithelium because there's more than one layer of cells. Well, how do we deal with this as far as cell shape when we've got more than one shape of cell? Appears to be the bottom ones are cuboidal or subcolumnar maybe. The top ones are obviously flat and squamous if you look at them from the side. The rule of thumb is you categorize shape by whatever cells are on the apical surface. This is the apical surface, and the cells here are flattened, they're squamous. So we've just drawn a stratified squamous epithelium. I'm going to draw one more picture, then we'll start taking a look at some examples on our PowerPoint slides of some of these different things and thinking about function, how these are related to function, where you might find these in the body, what they do. One more scenario, same orientation. Let's draw a basement membrane across it. And again, our other tissue down here, and our space apical surface will face this way. So same kind of orientation. This time we're going to have some cells that might look like this. Okay. And then I'm going to put some nuclei in here. Let's put these nuclei in. in each of our cells. When you look at this, perhaps through a microscope, you're going to see these nuclei, and they're going to obviously be at different levels. Nuclei tend to stain pretty dark, that's pretty obvious. It almost looks like you've got two rows or two layers of cells. It almost looks like it's stratified. It initially appears to be that way. But under close inspection, what one sees is they are not truly stacked one on top of the other. Instead, every one of these cells are actually all touching the basement membrane. They really aren't stacked one on top of the other. Maybe they don't go all the way to the surface, but they don't stack one on top of the other. Because of that, this is often given the special name pseudo-stratified. Pseudo meaning false. It initially looks like it's stratified, but not truly stratified. And because we've got some tall cells in there, we'll put the columnar as our shape adjective. So pseudo-stratified columnar epithelium looks stratified, but the cells are not truly one on top of another. They're not truly stratified. Questions so far?
Going back a little bit, how did you say that you defined the other one?
The stratified squamous epithelium?
Yeah.
Stratified is more than one layer of cells. It can be 2, it can be 5, it can be 100. As long as it's more than one, we call it stratified.
I was talking about the squamous
Oh,
part.
the squamous? Oh, okay. Why we had the squamous? When we have more than one shape of cell in an epithelium, it would be, we classify the shape by what's on the apical surface, what's facing the space. Good question. If I drew this instead like this, if I put some of these cells on the outside, we'll just make it 2 layers. If we did this, we made it really tall cells, we just made a stratified columnar epithelium. Because now the cells on the apical surface facing space are rather tall, rather column-shaped. Let's take a look at some of these different kinds of tissues. This is a simple squamous epithelium, so a single layer of flattened cells. In the top picture, we're looking at it from the surface, so you're looking at it just like this. On this one, you're looking at it from the edge. This whole thing is not epithelium, it's just this single row of cells right along the edge. Just likewise, the blue down here is not the epithelium, just the tan-colored area is this single layer of flattened cells. Function for this particular tissue, I'm going to write up here. This function actually is pretty complex for simple squamous epithelia. I'm going to write up here selective transport. For a lot of things, it allows materials to move from one side of the epithelium to the other with relative ease. For example, we have this kind of tissue in our alveoli of our lungs, in our capillary beds. It's what allows oxygen to move from one side to the other. It is so thin of a tissue, being it's flattened and a single layer, that it's a very short diffusion distance. So in one sense is that materials move through, some kind of molecules move through very readily. It's somewhat selective though because the molecules have to move through the cells. And the cells with their membranes and proteins have some sort of ability to control what moves through. So some things move through and some things don't. But lipid-soluble things move through very, very rapidly like CO2 and oxygen and some other things. But it can also be a fairly important barrier at times as well because of that. And we find these in lots of different areas of the body. We see some here on the outside surface of the gut organs, again, blood capillaries. There's places important in the kidneys. And some of this we'll mention later on how they function, these different organs, as we get to those individual organ systems. Simple cuboidal epithelium will be a single layer of cube-shaped cells. widespread in the body. Again, one place we see this is in kidneys. And a quick overview about the way your kidneys work. Well, kidneys take material from the blood, various molecules from the blood, and they're going to be exporting some of that, making urine. So kidneys is where urine formation occurs. As it turns out, there are a lot of fluid leaks into these kidney tubules. In fact, maybe 150 or more liters per day. Now, you do not urinate 150 liters per day. A lot of that gets resorbed back into the bloodstream. There's other toxin materials that are secreted into it in the urine formation to make the urine. So what happens here is we've got all of these kidney tubules. Now just imagine the epithelium is only this wide. Each is a different tubule, and each tubule, each little tube, has only a single layer of cube-shaped cells. In order to make this urine, sometimes there are materials that are resorbed or absorbed, and sometimes materials are secreted. So major functions here, and this is true for nearly all of simple cuboidal epithelial tissues. Absorption or secretion. We've got our cube-shaped cells, and then the tissue that's underneath here. Sometimes we have things that are being exported out that are being secreted. Sometimes we have things that are being brought in. Both of these are happening in kidney tubules, by the way. Secretion and absorption. Again, the cube-shaped cells may be a little thicker than squamous, but still it's just a single layer, and it's a pretty good anatomical morphological arrangement for allowing materials to move from one side to the other. Simple columnar epithelium. Now we learned already this would be a single layer of column-shaped cells. One of the common places this occurs is lining the inside of the gut. You think of the hollow inside the gut, particularly like small intestine. It's going to be facing into the hollow of the small intestine. Function for this tissue is pretty much the same as simple cuboidal. It's going to be involved in secretion and absorption. And to help understand that idea, we've got two more terms to know associated with this, terms that we might see in other tissues as well, but they help us to understand this idea of absorption and secretion. First of all, what might be being absorbed in the small intestine? What's brought from the inside of the small intestine into the bloodstream? Nutrients. Okay, some of you got a chance to eat breakfast this morning. You've eaten breakfast. It's probably moved all the way down to the small intestine. It's being digested. It's being broken apart. And those nutrients, the lipids, the carbohydrates, whatever that you ate, are being absorbed through these cells and brought into the bloodstream. To assist that, there are these little finger-like projections called microvilli. We haven't had that word yet. Microvilli are not cilia. They're not involved with moving things. Instead, what they do is they increase surface area, in this case for absorption. Again, think about our two scenarios. You could have a cell like this, or a cell like this, and we have absorption going on in it. This one has relatively little surface area on the top. This one has lots of surface area, which means that more nutrients can be absorbed. Your small intestine is about the size, at least in a living person, of a 6-foot-long garden hose. If you imagine a 6-foot-long garden hose, you split it open down the middle, that's not a very big surface area. Maybe half a square meter or less. It's pretty small, about that big of an area. Through the use of these microvilli and a few other anatomical structures, your small intestine has an inner surface area of over 200 square meters. That's around 4 times the floor space of this room. And it needs that to absorb all those nutrients. A couple of diseases that are involved with destruction of the villi and microvilli in the small intestine, like celiac disease, for example, that causes some problems about nutrient absorption, just general digestive properties. More about that when we get to digestive system in the second semester. We've covered absorption. How about secretion? Goblet cells are involved with secretion. Goblet cells, which are these paler looking cells here, secrete mucus. They're putting it into the inner part of the small intestine. Why are we secreting mucus in our small intestine? Well, it helps, helps to dissolve the nutrients. It helps to lubricate food as being mixed around and move through the digestive tract. Remember those old commercials about got milk, person's in a panic because he or she ate some dry cookies and can't swallow because it's really dry. The mucus does that. I forget how much we secrete, maybe 10 liters or so of mucus per day in our gut. Most of that we resorb, obviously. But you all know if you get some kinds of GI tract dysfunction, you can have very watery stools because the mucus doesn't get resorbed. Colon dysfunction, for example. Questions so far? Yes?
Okay, so the function of absorption and secretion, that's for all of the epithelium that you...
Absorption secretion is a pretty good functional description for simple cuboidal and simple columnar, especially for those. And that's pretty good. You've got other simple columnar in various other places in your body, and there's a lot of absorption and or secretion going on with that. Let's look at a stratified tissue now. We're going to see a different change in function because now we've changed the anatomy. This is stratified squamous. We have lots of layers. In fact, most of these are showing 20, 30, 50 layers in here, so lots of layers. The outermost ones, the apical surface up here, are flattened, so we're going to call it squamous. Now we're going to change our function to protection. It's a lot of layers. It's not very conducive to try to absorb or maybe secrete something through all these different layers. It's just too big of a distance. Diffusion wouldn't work very well. It would be very difficult to do. So it's very limited absorption secretion, but it is very protective. You have thick layers, that's going to protect. You can even erode some of the top layers off, just replace those fairly easily. Where do we find stratified squamous epithelium? The outer surface of the skin, the epidermis, is that. Near some organs with openings to the outside of the body. Vagina, the opening to the rectal canal, the anal canal of the rectum has this kind of tissue. These are areas that are exposed to pathogens, are exposed to maybe mechanical trauma, areas where you would need more protection. So now we've changed the anatomy, we've changed the morphology by adding layers, and that's changed the function. Changed the function a little bit. Questions? Hopefully you're seeing a little bit of how anatomy and function, physiology, are correlated. Hard to separate those two ideas. Stratified cuboidal epithelia are fairly rare in the body. There's only a couple places. Some of the sweat glands. Here's some glands associated with the esophagus. Not a lot of places where they occur. Again, more than one layer, and the apical surface is cube-shaped cells. Function for this tissue, it's kind of a little bit both of secretion plus some protection as well. It's not that protective. Why? In this case that you're looking at here specifically, why do you think this is not as protective as the previous slide?
Not as many layers.
Not as many layers. That's exactly it. More layers, the more protective. In your skin, the epidermis, you've got maybe 100, 150 layers of cells in the palm of your hands. You've got 20 or 30 maybe up on the scalp or maybe on your trunk where you don't have as much physical trauma. You don't need as much protection there. More layers, the more protective it is. Simple cuboidal epithelia are usually just in 2 layers, and so they can be involved with secretion to a certain degree. They're a little bit protective as well. Same thing for stratified. Oh, here's a sweat gland there. You can see the multiple layers at the base of the sweat glands. Stratified columnar, same thing as what we saw before, a combination of both protection and secretion. We find these in various places. This shows some glands. I know there's some in the pharynx, the esophagus, a little bit of the urethra. Not all of the urethra, parts of the urethra have this. Somewhat protective. But when there's just two layers like we see here, then secretion can be still relatively important as well. If there's a lot of layers and it's stratified cuboidal, then it tends to be more protective. Questions? Here's our pseudostratified tissue, and in this one what we see is nuclei that appear to be stacked up but are not truly stacked on one on top of the other. That is, they are all touching the basement membrane, and it's columnar, so we have column-shaped cells. There's some goblet cells in here, but there's something else that that doesn't make it pseudostratified, but that is present on this pseudostratified tissue. In fact, the only place I know pseudostratified is in the bronchi of the lungs, the trachea, and they all have nice, big, dark, obvious cilia. We can see it looks like a carpet right on the surface here, this cilia. Looks like you've got a clicker question, so I'll go ahead and let you respond to that one. We remember that cilia can move. In this case, they move materials over the surface of the trachea. One of the problems when you breathe in is dust and dirt and even dryness. The deep parts of your lungs are very sensitive to that. So you have a nice, moist, mucus-covered set of tubes. And that catches a lot of the dirt and the dust. And the cilia then constantly sweep that up and out of the body. That's what those cilia do. Let's see what you come up with here. What surface are they on this one? You guys picked here, you picked apical. Excellent. They are on the apical surface. If they are on the other surface, they wouldn't work. They have to face into this space. So apical surface is correct. I don't know if you if you know anybody that's a long-term smoker, or maybe you have a smoker in the family. One of the characteristics of them is that they often have a coughing spell early in the morning. Well, why is that? Well, one of the things that happens with the cilia is they get narcotized or sedated or even destroyed by toxins in cigarette smoke. So they are not very effective at cleaning out the lungs, and even mucus may be increasingly produced because of the dryness of the cigarette smoke and the actual irritants that are associated with it. So especially at night, a person ends up with, they're quiet at night, they end up with a lot of mucus deep down in the lungs, and they can't get it out with the cilia, so they actually have to kind of cough it up, and it takes them a while to do that. The cilia can't do it, so they have to cough a little bit more, or to get enough of an active process to get it up and out of the body. And you may not be able to see it from here. You can see it later when you look at the PowerPoint. This just shows nice, thick, carpety cilia here. This is increasing concentrations of the toxins in cigarette smoke that actually cause, at some point, actually removal of the cilia. And so you tend to build up those toxins. And that's indirectly related to, of course, lung cancer. And we can see that, again, here, brown is high rates of lung cancer, green is low. And so again, here I'm trying to pick out things that you guys are going to be dealing with. A lot of respiratory illnesses in East Kentucky. We have one of the highest rates of lung cancer, mostly from our high rate of smoking. As I looked recently, cigarette smoking has declined in every state in the U.S. except for Kentucky, which it's actually increased in, strangely enough. So again, you've got a long-term supply of patients associated with this. Oh yeah, there's high rates of, a lot of it is our high rates. We also have fairly poor air quality because we've got a lot of coal-burning power plants in our area. Even though it's improved, we have the poorest quality air in the nation right here, centered on kind of around Huntington, West Virginia, Ashland area. Again, because we have government incentives to burn high-sulfur coal in our area. And so anyway. Other kind of epithelium is transitional epithelium. This is our last kind of epithelium that we're going to talk about, transitional epithelium. Notice we don't have a layering descriptor. We do not have a shape descriptor. That is because shape changes and layer changes. This is an epithelium that's very stretchable, very distensible. It lines the inside of the urinary bladder. Now, the way your urinary bladder works is not like a bag or a bottle. It's not a fixed shape that slowly fills up with liquid as it's being produced. Instead, it accommodates just the amount of fluid that's there. So if you just urinated, it's about the size of a walnut. You know, it's a little bitty thing. It's all shrunk down right around that. If it's been a while since you've used the bathroom, it's quite large, maybe orange size or grapefruit size. It accommodates whatever amount of fluid that's in it. So it has to stretch. And this kind of a stretchable epithelium here is going to change shape as a result of that distension. So it may have lots of layers and they're kind of more cube-shaped or oblong or rounded. If it's stretched out tight, like down here, they may be fewer layers and kind of flatten. It kind of rearranges itself. It accommodates distension. It accommodates stretching, specifically adapted for that kind of scenario. As far as I know, this only occurs in the urinary bladder and maybe some very adjacent areas of the urethra. I'm not aware of another place where this kind of epithelium occurs. That's a good question. Yeah, its stomach is obviously greatly changes shape as well. One of the things about stomach is the whole thing is situated in some big folds. There's folds that are called rugae. So it's almost accordion-like. You could really expand the pleats out or contract it. But the surface of the membrane isn't quite the same as that. There may be some other adaptations associated with that as well. But yeah, it does stretch a lot, but in a little bit different way than the urinary bladder does. Oh, we've got our movie linked to the next topic here. And anybody remember what movie this is from? A little bit older now. Silence of the Lambs. And there's a famous quote Anthony Hopkins here says in this one. And it looks like it's a clicker question even. It's a movie clicker question. And let's see if anybody remembers this one, which is relevant to what we're going to talk about. While you're thinking about that, I need to...
Another family favorite of mine.
Ah, yes. This is a...
We should have a movie
Anthony
night.
Hopkins' signature role. And the books are quite good, I will tell you as well. I don't know if you've read the books, but they are quite good.
Have you seen the series? I don't even see. I know.
Especially, I forget the writer's name, but the last one he wrote out of the three was actually Silence of the Lambs. He gets steadily better, and the last ones are pretty good. Or maybe that's the second one he wrote. I can't remember. At any rate, let's see what you picked on this one. Strange eye clicker question, but nonetheless, a few of you apparently are fans of this one and know it was liver. Well, yeah, liver. And it turns out liver is a gland, and glands are made up of epithelial tissues, or at least mostly of epithelial tissues. So let's talk about glands just for a couple of minutes. We often distinguish between endocrine and exocrine glands. They're both made up of epithelial tissues. They both produce something. How do they differ then? Alrighty, let's make some more drawings. We often can learn by drawing. It's a way of practicing that's active. And I'm not going to draw any specific shape, but we're going to draw a bunch of cells. These are supposed to be cells. Whoa, I have trouble with holding on to chalk, don't I? This is going to be exocrine here first. It's an exocrine gland. And here is more of the glandular portion right here. It's making something. It's secreting something. The material that it produces travels to its target destination by way of a duct. There's a tube, a duct. Imagine like a sweat gland that has a duct leading to the surface, or a salivary gland leading to the surface. The pancreas is mostly exocrine. It has a duct secreting digestive enzymes to the small intestine. We would also consider goblet cells, which are unicellular glands this way as well. They just go directly to the target area in that case. And that's exocrine. So things that might be produced by exocrine glands, mucus, saliva, digestive enzymes, a number of things like that. Sweat. In endocrine glands, there is no duct delivering the produced product into the target area. There may be a little hollow there. They're secreting whatever they're secreting. So how does it get to its target destination? It does so by way of the bloodstream. That is, the blood picks up the particular material being secreted and delivers it to its target destination by way of the bloodstream. There's no duct involved. We call these endocrine glands, and the kinds of materials that they produce we generally call hormones. Messenger molecules that are produced by glands and delivered by way of bloodstream, we use the word hormones. Some kind of glands have dual functions. Pancreas is about 99% exocrine producing digestive enzymes. Small amount of it also produces things like insulin, glucagon, hormones, which arrive via the bloodstream to destinations. See some examples of endocrine and exocrine here. Here's a skin with, you can see a sweat gland. I've kind of outlined in black a bit here. The main secretory portion of the gland is down here. You can see a little bit of a duct leading up to the surface. That's an exocrine gland. So that's a good example of exocrine right there. Questions?
Can you repeat that last statement?
Yep. Well, the sweat gland, this is an exocrine gland.
Okay.
The secretory portion of it is down here. It looks like a big garden hose in a knot. We'll explain more about how sweat glands work later.
Okay.
But it produces its product here, and there's a little duct that leads up to the surface. So and that's where the sweat is discharged onto. So that's a good example of an exocrine gland.
Okay, thank you.
Uh-huh.
What did you say the function of the
Pseudostratified. I guess I didn't say a function, but let's see here. There is some secretion associated with it because of the goblet cells. And maybe I might even say transport because of the cilia associated with it. They're transporting mucus, maybe even cleaning, that very specific kind of thing. Again, pseudostratified tends to be restricted in location, but nonetheless an important tissue. Now for connective tissues, we're going to have enough time at least to introduce a little bit general about connective tissues. How are they different from the previous ones? Well, we already talked about a general thing that they do. They tend to bind, that is, attach one thing to another, support, structurally support, physiologically support, strengthen. That's their general functions. In contrast to the previous one, they have a lot of matrix. Lots of matrix. Matrix is composed of two things: fibers and an amorphous material called ground substance. So if we look at our connective tissue here, we see there are cells, and the cells are much more widely spaced. Notice they're not all touching one another. Maybe different kinds of cells widely spaced. So lots of matrix, tends to be lots of extracellular material. We have these fibers, different kinds of fibers that run back and forth in these. And then also in between the fibers and the cells is ground substance. It more or less takes the shape, fills in, packs in whatever space is available. This ground substance may be liquid, as in the plasma of blood. It may be gel-like or rubbery, as in cartilage. It may be mineralized, as in bone, a hard concrete mineral. But it's kind of the filler in between all the cells and the fibers. We'll have more examples of what ground substance is like when we get a little bit further on. There are a couple of different kinds of fibers that are present. Two of these I'm going to mention that I'd like you guys to be familiar with. Collagen fibers and elastic fibers. Collagen tends to be very strong. Very strong with but not very stretchable, not very elastic. So like a rope or a chain. So they're pretty strong, but not very stretchable. Elasticity by definition is the ability to recoil to regain its shape once it's been distorted, are often relatively stretchable, but then when tension is released, they regain their original shape. They have good recoil properties. These would be more like a bungee cord. So very stretchable, but will retain their shape once the tension's let off of them.
Still
And
strong though.
they're still pretty strong, yeah. Still pretty strong. Maybe not as strong as collagen fibers. And what we're going to see in the future, we're going to see that by changing the orientation of these fibers, the kind of fibers, the amount of fibers, one imparts different qualities to the tissue. Now, also differing between this one and epithelial is that usually, not all the time, but usually connective tissues are highly vascularized. Oh, it looks like a reminder. Do you remember what highly vascularized means? So epithelial is the last thing we're going to do. Epithelial tissues are, again, not vascularized. Connective tissues are vascularized, usually. There's a couple of important exceptions to that. Connective tissues mean they're highly vascularized, meaning they have good air spaces, good blood supply, good nerve supply. What does that refer to? You guys selected on this one. You select here. You selected B. Okay, most people remember this. Yes. So it means they have good blood supply. Not all of them do. There's a few exceptions, but most of them do. Okay, we will stop there.